Yeah.
Oh, so Florida's in the final four, is
Yeah,
it?
we made it to the final four.
Wow, that's a big deal. Oh my goodness, I didn't know that. Excellent. Good luck.
Yeah, nice.
Yeah. Yeah.
Pass it on to them.
All right.
All right, over
Dylan?
to you, Dylan.
Yeah, go
Okay,
ahead.
so again, I'm Dylan. I've been in Angie's lab since I guess 2020, right? And just just for Clinton. And so of course Angie suggested that I look into control strategies for limiting crypto, right, once it gets into the host. So as you saw in that email, I found that paper, and if I can share really quickly a little presentation that I put together, um, so you can see this here. So can you all see my presentation
Yes,
now
looks
for the
good,
Windows
Dylan. Go.
for it?
Yep.
Okay. So just to go back to the beginning, so uh, they made the synthetic oxytroph, right, using and Saccharomyces, and they were focused on industrial applications. So they had their synthetic oxytroph that could only grow when it had O-methyltyrosine present. And of course, how would we apply this to Cryptococcus if we wanted to use it for biocontrol? So I've kind of put together an outline of the steps. But maybe before I begin, do you have any thoughts on the paper? Should I go back a little bit more over it, or talk about some of the figures in a little bit more depth, or am I good to kind of keep going with like how we would implement what they did in Cryptococcus?
I think I'm good with the paper. Yeah,
Okay.
you can move ahead.
Okay, great. So I guess just point by point, we'd first decide on an essential gene to target. So a gene where we would make the mutation. So we might decide on that based the on either by its expression or just out of a list of known essential proteins in Cryptococcus. And then once we've identified that protein, we look for which positions within it are tolerant of mutation so that you still get the normal function of the protein. And they outlined use a SIFT algorithm, at least that they mentioned, or they had some other approaches for identifying those positions. Then we would change that position to to stop codon and then express their the Lu Omer RS and tRNA pair. Uh, and we could do this into a temperature sensitive mutant potentially to down the line increase the control. Um, and then of course we'd isolate the synthetic oxytroph, and then we would hopefully determine the minimum concentration that they needed. I think they had, I believe it was zero point 0.2 millimolar was their concentration, or I think they showed growth. They tested down to 0.02 millimolar, but probably different Cryptococcus and depending on the gene. Um, and we'd want to, after we have that strain and determine what concentration is permissive for growth, we'd want to measure the escape rate, and maybe do some other in vitro assays potentially, to make sure that it still has the wild type phenotype with respect to barrier crossing or anything else we might be interested in. And then we want to decide at that point whether we want to pursue other strategies like the ones that they mentioned, and maybe adding we decided one gene here, knock that out, use it to uh, cause the synthetic oxytroph phenotype. Maybe we could do a second one to increase the control, or we could add the transcriptional control that they talked about somehow, the changing out a promoter somewhere, maybe to something that could be inducible with something else that could be loaded into the nanoparticles. And then of course we'd once the strain's ready, hand it over um, and do the nanoparticle based assays that you do. So, so does that make sense? As kind of roughly, if we wanted to make the strain, how everything would fit together.
Yeah, so a, a couple things. Yeah, for the most part, Dylan, it makes a lot of sense. You mentioned temperature sensitive. I wasn't sure why we would need a temp sensitive mutant. Was that
Um,
just based on selection or?
I think so. I don't think it's for the selection. I know that they used the temperature sensitive mutant when they created their strain. I was more thinking of the temperature sensitive mutant just for better control. And so it would be a non-dividing at at temperature. It wouldn't be a temperature lethal mutant, I
Okay.
think.
Gotcha.
Something like the RAS1 strain, just to add an extra layer. So if there was some kind of escape from the synthetic oxytrop, it still wouldn't divide. But I think that might be optional, whether we'd want to use the temperature sensitive mutant or not.
And then I think with respect to additional strategies, the bottom line here is that, we want to be able to sort of develop either therapeutic or maybe diagnostic in our nanoparticles. So we
Mhm.
kind of want to keep the amount of nanoparticles we need to load with sort of this inducible drug to a minimum, or inducible molecule to a
I
minimum.
see. Yeah.
So, if we can get this working at a lower
Yeah.
concentration, required concentration, lower threshold, as well
So
as reduce the number of particles that we have to sort of put on the surface that contain this inducible molecule, then the better.
yeah. So I think that by targeting multiple genes, we might be able to achieve more with less. Also, I think that um, they're concerned about escape. I actually have a slide on this if I can just switch to that. Um, so they were concerned about whether, let's see, so we might be able to tolerate a lower rate of escape, right? So we might not need the backup strategy because they were looking at up to 12 days and in very large fermentation conditions. But if we're only concerned, I don't know what the inoculum size would be to deliver your nanoparticles, right? But if it's less than the 10 to the 9 cells that they are concerned with here, if they're looking at an escape rate of 10 to the 9, if we're only introducing 10 to the 6 cells, we might not need to have a backup strategy. And also if we're only looking at, 4 days or something like that, it might not be necessary to do this two-layered approach. Um, so I'm not sure at this point whether we might just need to knock out one gene, and we might not need any of these backup strategies anyway. But it'd be good just to assess once we have the strain whether it would be necessary. Um,
Yeah,
so that's
I think we start with a simple strategy. We're likely going to be injecting a lot less. We also have the immune system to back us up, eliminating these bugs. So,
mhm, that's great. Yeah, yeah. So but you can see here when they did the two genes, they still had no escape even with all the cells they were using. Um, okay, so I went ahead and tried to do a little bit of the first step here of identifying a target protein. And so they targeted, for most of their paper, they're talking about CDC4 and CDC27, with genes of that name with roughly the same function do exist in Cryptococcus, but I looked into it. They're not homologous, but I decided to go with it anyway with CDC4, just as kind of a trial run to see how you would actually go about selecting it. And fortunately, the CDC4 gene in Cryptococcus, um, it is, let's see, it's supported by the kind of algorithm that they talked about for identifying which sites you can make a mutation at that are tolerated. So where you could switch in the stop codon. By the SIFT algorithm, Cryptococcus is one of the 200 organisms supported by this. So that was kind of nice. Um, and so how it works is you select your protein, you feed it into the SIFT algorithm, and it gives you all the sites that might be tolerated. But I like at the beginning, I just want to point out that they picked so many different genes and at all different transcription levels that I think I mean I think we have a lot of genes that we could work with potentially, just any essential gene. Um, but that's all I wanted to point out with this slide here. This is just their supplementary figure one. So anyway, moving on with how you actually would go about selecting a site. I'll just do this really quickly, just to make sure that I understood the tools that they were using. You can load in the SIFT algorithm, and then you give it will give you an alignment with closely related species, and then the substitution rate at each site. And so I identified some sites within the CDC4 Cryptococcus that you could potentially make a mutation at. So I guess anything above 0.05, like this is at 0.51, this position, um, would be a candidate. So anyway, or I don't think I did this. This was preliminary. I don't think I did this quite right, but I used Swiss Prote rather than what they used, the cluster NR database. So I think, and that I think would give more hits using the cluster NR. But the point is, anyway, I could do this really quickly and identify some sites, and not just with this gene, but with any essential gene, I think. And then I tried it another way too, using some different tools. So the problem with this here is I identified sites that were not quite at the N terminus like they talked about in the paper, but doing it this other way, there could actually be some closer to the N terminus, but I don't know if that's necessary. So anyway, I generated this alignment using other tools, and um, I think anyway we have some sites here if we were to go with CD40, the CD4 in Cryptococcus that could potentially introduce this stop codon at. So anyway, that's all to say that the kind of analysis that they did in terms of how to select specific codons to make this insertion at, that's doable in Cryptococcus, just like it is for them in Saccharomyces. So are there any questions about kind of this step of the that we go about doing this?
Dylan, can I ask how many mutations did they make?
They made a bunch. I mean, I have their
Did they?
here's their list. I don't know if you can see their list of plasmids here. So all of these here
Oh,
would
wow.
be the CDC4 and
Okay.
CDC27 that they made. So each one of these would be different. So just I don't know, several, and these aren't even all the genes that they targeted, but they made many mutations. I think they say somewhere in they claim towards the end of the discussion that you can go from having a target to do the substitution and to having a synthetic oxytrol strain in a week. I mean, that's
That's
their
great.
claim, but I think that they tried so many different strains. So this again, they're not in any way like locked into the CDC4. We still have to figure out which ones would actually be essential, which would be good candidates, which are high or low expression. We might actually want to pick low expression. I was thinking because if we pick a low expression protein, that might mean that we could use less of the synthetic amino acid. You
Yeah.
know, if
Yeah.
there's a lower copy of it, then you don't need as high concentration.
Right.
But we'd have to look into that. I don't know what CDC4, what its relative expression level is in Cryptococcus. I think in their figure 1 here, we have their CDC4. It's anyway, so in Saccharomyces, the CDC4 and CDC27 have relatively low expression on this curve that they have,
Mhm.
right? In terms of I think this is their transcript levels. So it's doable with those are their primary examples. So I think we could pick a couple of low expression.
Do you know what those genes are responsible for, Dylan?
That's cell division. I'm not familiar. You know, I don't work with CDC genes, but I believe they're responsible for cell division, cell cycling.
Yeah.
Yeah,
Um,
Yeah.
CDC24 is one of the gene knockouts that's used for temperature dependent growth. So I think it is along the same like family in terms of function.
CDC24.
Yeah, CDC24. We have that strain here in the lab.
We have 24 or 42.
24.
Okay.
At least that's what's written on the vial.
Okay.
Oh, yeah. Yeah.
yeah,
Yeah. Right.
I don't think it would, but I'm sorry, Andrew.
No, I was just going to say it's doable, but Kim is right. It certainly isn't like Saccharomyces, right? So it will take a little bit longer, but I do think it's doable, definitely.
Yeah. So Kim and I have been talking over the I guess since Thursday or whenever about different how we would actually go about doing this. And so I we're looking a little bit, and Kim tells me he sent me this article from a while back um, so in their what they used were centromeric plasmids, I guess, to get longer term stability inside of Saccharomyces. And Kim tells me that there's similar plasmids available
Crypto.
for crypto, or we could go with other approaches like CRISPR, but I really defer to Kim on kind of the details.
Well, hang on. If a plasmid is centromeric, it needs a selection, right, to stay. Otherwise, it'll get kicked out. Um, or you could integrate it into the genome for long-term stability, right? I know that Jenny Lodge, right, Kim? Is it Jenny Lodge that created that episomal plasmid for crypto? And we were trying it once years ago, but we never went back to it. Yeah, to try it. Yeah. In the paper, was it like an episomal plasmid that they expressed in Saccharomyces? It wasn't integrated, right?
I don't think no, they just
Yeah,
they introduced the centromeric plasmids.
yeah.
Just they introduced them both to make just both to introduce the tRNA and also
Hmm.
to make the actual to do the homologous recombinate to make
Right.
the codon swap. So they use centromeric plasmids for both. Um, but I think I have to read into that detail a little bit more about obviously with the homologous recombination one where they're making the codon swap that becomes integrated, right, or the change
Yeah, yeah.
permanent. But for the other one, I'm not certain. I guess they still kept it in their fermentation under selection, maybe.
Yeah, yeah. They
But
use the
I'm
Gelt
not.
promoter. Yeah, yeah. Yeah, we have all these plasmids, we have all of them. I don't know if they're going to be useful to us, but we do have them. But anyway, yeah. I think, Jamal, I had a question for you though. I mean, I think, doing the modification in crypto, I think is doable. The question is whether or not you can carry the synthetic amino acid the way you have it in your nanoparticles. Is that going to be the bottleneck here? Is that going to be the difficult part of this whole thing? Because I think making the mutations in crypto is doable. I don't think that's going to be an issue. But what do you think about this idea of having to carry or add this amino acid to your nanoparticle?
Um, this is what was the size of the amino acid again? Was it just
It's just the one. Just the it's
a
I
couple
can I
daltons?
don't. Let's see. I have the MSDS up somewhere here. It might actually.
It can be that big. I think we could fabricate particles with anything,
Yeah.
to be quite honest. Which is why I think this is worth a shot. Now, the question is going to be how can we sort of maintain local concentrations of these amino acids for, a couple days as it gets into the brain, hopefully, or elsewhere. That's going to be sort of the challenge. And that's, I think, just going to be a materials challenge more than anything else.
Okay.
But I think this is quite feasible,
So
especially to keep those concentrations to very, very low
Yeah.
order. Yeah, it's definitely feasible. We can, yeah, we can formulate to accommodate those conditions.
Hmm.
so they use 1 millimolar in their initial test, but
Sorry, I meant what was the molecular weight of the amino
oh
acid?
so it's hidden for me behind the screen here. 1 second. That's not what I wanted to show. Let me see. I'll just look up the MSDS for it for O-methyl.
It doesn't matter. I mean, it should be pretty small. It's an amino acid, so it should be
Yeah.
pretty. So
Should be.
we can load that a couple different ways, and it's again going to come down to us sort of playing with the material properties so that we can get the right release profile to keep um, these things alive, or at least keep local concentrations around these things to above a particular threshold.
So they mentioned that it's membrane permeable. So I don't know if that means something about its chemical properties.
Yeah,
I
it
don't
just
know
means
if
it
that
can get through the membrane without without needing sort of extra assistance
Yeah.
from channels or whatever.
And so those should, you can still, so if it's, I mean, it can't be like lipophilic or something, but you can still load those into the nanoparticles just fine. So I just wanted to point out with this slide here is they were concerned with maintaining growth rate. I don't think we need to be concerned with maintaining growth rate. So they showed that you could go down to, they said down to 0.2 millimolar for full growth rate, but you can see it still grows fine, just more slowly for this particular mutation. This is for their CDC4 mutation, and no guarantee it would be the same curve if you mutated the Cryptococcus CDC4. But you know, if you're willing to accept a lower growth rate, I think you can also get down to quite low concentrations, which, you know, we're not doing large scale fermentation, so we don't need.
Yeah, I mean, honestly, we're looking at if we could actually limit that growth rate, it will be fine. We just want to keep this thing
Yeah.
alive more than anything else, right? For a short window, and then have them die. So if they're not proliferating, that's actually beneficial.
Yeah, which is just like we don't need to. We don't really care about the growth rate.
We do because we'll have to show these things, but
Okay.
yeah. Yeah, it's pretty small.
Yeah.
Let's see. So, you know what? I think we've almost. I just have this one last slide. And actually, I'm sorry. I had the cast here. I guess it doesn't have the molecular, but this was just to point out that it's not recognized to be hazardous, so it should be fine from that perspective. I mean, especially if you're using at very low concentrations. And also if it doesn't work for whatever reason, they do mention that there are potentially other options. Obviously, be more work and kind of venturing into uncharted territory. But if O-methyltyrosine doesn't work for some reason, I think maybe we could try something else too. I'm not sure, but they seem. I mean, obviously they're trying to sell it as an easy swap. I'm not sure if it would be, but I think that there are other options, and I don't think we'd have to stray from O-methyltyrosine anyway. So that's it in terms of what I had for my little slideshow. But yeah,
Okay.
just to recap, or just to go back to this slide here for reference.
Quincy, you had a comment?
What's that?
Well, I was thinking in terms of like how we would integrate the amino acid. Right now we're thinking of having nanoparticles that encapsulate the amino acid and then gradually release it. I was wondering, or I was going to comment, there might be an opportunity. Let's say for whatever reason we tried the intracellular hydrogel to also like prevent the replication. You may be able to encapsulate the amino acid within the hydrogel, and then it's already inside of the cell, and over time it could just diffuse out. That was more so thinking out loud. In terms of another way we could integrate the amino acid. Because if we have the surface bound nanoparticles, it would have to release and then.
You want to expound about on that? So it's another idea, right? I'm hoping that it's something we don't have to pursue, but it's an idea that would probably increase the complexity a little bit. But you can perhaps educate the group on that idea.
Oh, sure. So
this is an idea coming from Chimeng Tang's lab. Angie, I don't know if you know Chimeng.
Sounds familiar. Here at Davis?
Yeah, he's in BME at Davis.
Okay, yep, go ahead.
Yeah, so his group has come up with a way to basically prevent microbes from dividing. It's a synthetic biology approach where you can have microbes that basically perform a function, but they don't have that biohazardous component to them because they integrate a hydrogel into their intracellular environment, and the stiffness of the hydrogel basically prevents the cell from dividing. So they are able to establish some sort of biocontainment
That's
that way
interesting. Is this in
well.
bacteria?
They've done it in bacteria and in yeast, and I just saw that they submitted a patent for certain bacteria and certain yeasts where they've
Interesting.
implemented this intracellular hydrogel. So that's, I think
That's what you
Dylan
were getting
mentioned,
at. Right.
yeah, Dylan mentioned like having the backdoor safeguard in case the tyrosine doesn't necessarily like prevent the replication. We also have that
There's
option
another way to
as
do
well.
That's interesting. Yeah.
it. Yeah.
So,
I think
so
there were some caveats to that, the Clinton, right? I think I can't remember exactly what was the challenge with that.
Well, yeah, the caveat is that they use, or one of the ones that I identified in the journal club was that they use UV irradiation to um, create the
Awesome.
hydrogel, which could ultimately damage the CN. But based on the monomers that they used, I think we could just opt for like a different photo initiator that doesn't rely on UV light. Like just visible light works for the type of hydrogel that they used.
So, I was also wondering about the capsule itself, Cryptococcus. You know, is that a backup? I mean, I wonder if you can put like, can you load more if this amino acid can will it persist inside the Cryptococcus capsule? You know, in addition to the
There's a little bit of
nanoparticle, maybe.
background noise, and so I can't quite hear you clearly, Dylan.
I'm sorry. So, I wonder if this amino acid, the O-methyltyrosine, if you can make the mutant, if it can be loaded into the Cryptococcus capsule as well. You know, maybe you don't need to take up all, because you're worried about, if I'm understanding you correctly, you want to reserve as much of the nanoparticle, right, for the therapeutic. So I don't know if the O-methyltyrosine, if it would stick around in the Cryptococcus capsule, or I don't know. It would be interesting to test.
yeah, I don't know. What do you think, Jamal?
I'm just trying to think of what sort of mechanism where we could load it into the capsule and still have it be released at a sort of
Yeah.
engineered, consistent manner to sort of keep these things alive for a short, sustained period.
I don't think that'll be too much of a concern because with the nanoparticles being attached to the surface, presumably to the cell wall and the capsule, if you're having the amino acid release, it could release in several directions. It's not like a unilateral direction. So some of it could release like directly towards the surface of the cell, and that way it has a direct interaction there.
I agree with you, Clinton, but this is a small molecule, right? So before, let's say, okay, so what I'm saying is that we have to figure out a way to sort of fetter it or localize it within that capsule. And if it's something as simple as absorption, then, as soon as it's an aqueous media, it could all just
Yeah.
diffuse
Be
out.
released, right?
So yeah, that would be a challenge. And I think we have probably a simple method where, if we can put, a tenth of the particles loaded with this drug, and then the other 9/10 be loaded with a therapeutic drug or diagnostic or whatever we're shooting for, then we're golden. Oh, okay. I see what you're getting. You're getting a targeting.
Yeah.
That's actually a pretty good idea. Um, I've had thoughts about it, especially since it's looking like with Clinton's work that CD44 is involved.
Mhm.
but we haven't incorporated it into the particles yet. So we're keeping this relatively simple now, using a particle that we know the Lewis kinetics pretty well for. We're gonna change it slightly, but yeah, there are some thoughts there as well. We just haven't explored it as yet.
So, Jamal, can I ask you just a conceptual question here? I'm having trouble visualizing this. So when you talk about the particles, are you, I mean, is crypto sort of encased in these particles, or the particles are attached to the surface of crypto?
Quentin, you wanna maybe give a quick presentation?
Oh, that would be
Uh,
great.
sure. Actually, my
Sure, let me
poster is up right now.
Impromptu.
Oh, a second. It's been so long since I've there.
Yeah, Dylan, if you make
Okay.
Clinton host,
Yeah.
yeah.
I think I got to pop up here. So hope, because I haven't done this in so long. Let's see. I'm sharing my screen. So if I stop share, then you should be able to share.
So
Right?
Andrew,
Okay.
the quick answer is that we basically cover the surface of the crypto with these particles.
With these particles, you surround crypto with them. Okay, okay. Yeah, we talked about this. I'm sorry. I remember that now. Okay.
Yep. So the idea is that we leverage some adsorption interactions where the particle can easily attach to the surface.
Okay.
So this is like just the cartoon version, but I do have some microscopy images
Okay.
showing the overlay of the cell wall and the nanoparticles that are surface bound.
So do you think it's binding to the capsule and the wall at the same time? Is it sort of getting embedded in there? Or are you
So
specifically targeting the wall?
we haven't looked that granularly yet, but we do believe that there's a possibility for both interactions to take place. Actually,
Okay.
I'm having one of my undergrads look at acapsular CN
I was
this
just
week.
gonna say, yep, you should try some ACAP strains. Yep.
But I have shown like through a very quick experiment that I can like attach the capsular antibody and show that there's co-localization of the antibody and the nanoparticles. I
Okay.
think something like SEM would be able to show us more clearly where the nanoparticles are actually attaching.
Okay.
But theoretically, they could attach like with the acapsular strain directly to the cell wall
Yeah, to the
as
wall.
well.
Right. 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 Okay.
So you're using the PLGA nanoparticles.
Yep.
Do you know if that's the same protocol that my undergrad Adam primarily was working with NOAA to produce the PLGA nanoparticles? I think he was using I only got a quick glance at that. That was the branch, the forearm PLGA nanoparticles. Are you still using that for your as the
No.
monomer in your, or you?
So I do remember when you and Noah were working together, at least through his slides, he was working with the forearm PLGA. We're using single arm
Okay.
PLGA. It's much simpler.
Okay.
So we used the forearm because we wanted to sort of have more handles to tag
Yeah.
your peptide.
That's right.
All right. So we can buy this molecule and probably start loading these particles and start working on the race kinetics, sort of tailoring the race kinetics so we can get consistent external concentrations of the molecule for 3 to 5 days, I would say. So
And
yeah,
yeah, on our end, we can try, Dylan. Do you want to try to make the mutants?
Oh yeah, definitely. I'd love to try to make the mutant. We'd have the obviously the biolistic or the electroporator
Yeah.
this area here. We have the materials. We have whether we need to figure out whether how exactly we're going to make the integration and make the list of what genes to target. But I'd be more than happy to start doing that. And and obviously working with Kim as well, try to identify candidate genes to make this amber codon insertion into.
so the lowest that showed in the paper was 0.05 millimolar. Was
They show for the concentration of OME and Y, and
yeah.
they showed down to Uh, so they have their two graphs here in their supplement. That was, I think, right. It was 0.05 was the lowest, but yeah. So I can bring up the slide, but I have the other graph here as well. So I don't
All
know if you
right.
can see that's 0.05,
Yeah.
that curve there. And I think you could probably go
0.01.
somewhere
Yeah.
like that would be fine as well. You could
Mhm.
go below 0.05.
Yeah. Yeah.
Okay.
Yeah. So let's see. Yeah, I think it's super interesting and something I'd love to work on. And I'd
Yeah.
be more than happy to devote um you know time and effort into making this work on my end.
I mean, it is worth a try, I think, because if it did work, it could potentially, Jamal, it could potentially be a way to control crypto right after you inject it in vivo.
Yeah.
And that really what it's all about, right? Kind of do its thing, deliver its cargo, but then you want it to go away. So,
Yeah. So the other two I guess design parameters that we would have, Dylan, is that it's still able to sort of be phagocyte toast, vomocyte toast.
yeah, we'd have to check all of that
Yeah.
to make sure that it doesn't lose any other characteristics. Yeah,
Already. So
we'd have to
um
confirm that.
most of what I've been doing here is, I kind of specialize in doing the in vitro assays in terms of barrier crossing. So I
Mhm.
think for me, that's almost the easy part is making sure that it still has, before we try to do in vivo studies or anything, taking these in vitro strains and checking them for crossing and making sure that they still might have some of the desired phenotype. I
Yeah.
think, yeah.
And in the past, we've done studies with macrophages just to show whether they get engulfed or not and look at all of that as well. Those are in vitro assays we could do as well.
Okay.
Yeah.
That's good.
So we've done that. Of the amino acid. Yep. Yep. Yeah, I agree. Hmm.
Um,
So do we want to just maybe touch base again in a month and see where we're at with
Yeah,
each end of
definitely.
the project?
I think that's reasonable. Does a month, is that a reasonable time frame? Uh, Dylan? Yeah. Yeah. Okay.
I can make a list I'm more than happy to kind of within that time, figure out what the target genes might be and also work out a better strategy for making the mutation. Maybe we'll even get to setting up the biolistic or whatever
Yep.
other pieces we might need to.
Yeah.
Yeah, yeah, we don't have to be done, Kim. Just touch base. Could be a quick 15, 30-minute meeting, just to touch base and sort of, make sure we're on track. No, no, no.
No, no, no. It might take a little bit longer than that.
Yeah.
But as long as we have a path forward, right? We've identified some genes and we know how we're going to go about it. I think that would be good.
Yeah.
Good. Dylan, that was a great find.
Yeah.
Excellent.
Thank you.
Yep. Very cool.
Yeah. I mean, I think so. Originally, when you just to give some background, I think Angie asked me to look into it, and I was So we have this urease knockout strain as well, and I was interested in using that strain, and then we can actually express urease in Pichia and use that. And so that was my original idea, was use the urease strain plus adding urease to the strain. But I think that this obviously much easier. So Angie pushed back on that, and that's why
Yeah.
I took another look and found this. So
Yeah, I think this molecule is a lot easier for us to work with too. Like
Mhm.
we can definitely find a profile of material that should release to desired concentrations. So
Yeah, that's great. All right. Well, thanks, everybody.
the
Nice meeting you,
conversation.
Clinton. We'll
Yeah, it's nice to finally meet you, Dr. Jelly.
yeah, nice to meet you too. All right, take care, Jamal. Good luck to the Florida. What are they? The Florida
Florida Gators.
you're right
Gators.
across
The
from the
Florida
stadium, aren't you?
Gators. Yeah, because you're in Gainesville. Are you in
Yeah,
Gainesville?
yeah.
Yeah,
I'm
you're in
right next to the stadium
Gainesville.
actually. Yeah.
Wow, that's
I did my
exciting.
undergrad in Florida, not at
Oh, okay.
UF, but anyway. So
You went to UF for undergrad?
no, I went to UCF actually for
Oh,
undergrad.
UCF. I see. Okay,
Yeah,
okay.
yeah, yeah. So kind of a
Yeah.
lesser
Let's
reputation, especially when it comes to football. So yeah.
cool. All righty. Well,
All right.
thanks, everybody.
Thank you. Thank you, Angie. Thank
Thanks.
you, Kim. Thanks, Dylan.